Good afternoon. Today, I would like to speak to you about the pituitary gland as a critical component of the hypothalamo-hypophyseal target organ axis. This is an important concept in the realm of endocrinology, which, as you all of us know, rather, that endocrinology is the science of endocrine glands and their secretions, which are known as hormones. In fact, the word hormone itself means to excite. And therefore, it very clearly implies that the secretion of the endocrine glands have a role to play in eliciting response for critical metabolic processes and physiological activities. What I plan to do is take you through a journey of understanding the anatomy, the gross anatomy of the pituitary gland, a little understanding of its cytology, a little bit about the mechanisms of communication between the hypothalamus, the hypophysis or the pituitary, and the target organs. And maybe we sum it up by understanding some important hormones of the pituitary gland. So to start with, in the first place, at what point of time in the evolution of life, but of course the fauna, did the endocrine system really evolve, and why? When there already existed a sensory system or a nervous system, what was the need felt to invest in yet another discrete endocrine system with its own glands and secretions? The purpose is very simple. And as you can see on my screen, when we speak about the nervous system, essentially that's got to do with sensory interventions on neural circuitry, which normally operate through electrochemical signals, which means, as most of us would be familiar with, the signal is conducted by a neuron with electric charges sweeping all along the axon until it reaches the axon terminus, which is where you have a very interesting molecule known as neurotransmitters secretions packaged within synaptic vesicles, which get ruptured and probably cross through the barrier of presynaptic membrane into the cleft and the target sites. But there is a limitation, and the limitation is that, first of all, of course, the good thing about sensory intervention is that they're very fast, which means they impart a great survival value. You want something to happen very fast, then the best way to control that activity is by putting into service the sensory interventions, because it's fast. The effect is very fast. But if it is about how long does the effect last, the answer is the sensory interventions normally have effect which is very short lasting. In contrast, if you look at the endocrine control, which is affected by the secretion of endocrine glands, which all of us know are known as hormones, the hormones act a bit slow. Sometimes it may take a few seconds and quite possibly even some days, as against the time taken for a sensory intervention to elicit a response could be a few milliseconds. But that which the endocrine interventions bring about may take a few seconds or a few hours, but sometimes even days. I am trying to say that the response elicited by endocrine interventions are slower in comparison with those by sensory interventions, but on a positive note, the effect is long-lasting. I mean, once the effect is elicited, it shall last for quite some time. I think that's the advantage. And unlike in case of the nervous system, where you have a major emphasis on control, an endocrine system is a combination of both control and coordination. So it ensures that many critical activities happening in a body, many metabolic processes, many physiological activities happen at the right time. They manifest into desired effects, and they synchronize with one another, which is that they have everything that's happening in a body has to be in sync with each other. I mean, I always give this example of an orchestra where you have a number of musicians playing different kinds of instruments, each one a maestro in his or her own, domain of expertise for that given instrument, but they all need somebody to conduct that orchestra so that they sync with one another. And if each one starts playing a tune of his own, then we eventually end up not with music, but with noise. And I think that's a good analogy, that in our body, there are so many things happening, all of them together. And if they have to manifest into some positive effect in terms of maintaining a constant internal environment or milieu, or what you call as the fixity of our internal milieu, homeostasis more technically, keeping the physiological activities at their optimal ranges, ensuring that everything in the body is happening at the right time, at the right pace, and by optimizing resources, then you certainly need a proper conductor, or in other words, a proper coordination, which is possible only with the endocrine glands and the hormones that they secrete. So I think that's the crux of the whole discussion. Today, we're going to focus more on the pituitary gland, which is also called as the hypophysis. It's a very tiny structure, not very accessible even for medical procedures. It's something which is smaller than the smallest of the fingernail, weighs very little, except when we talk about the pituitary in a pregnant subject, which is slightly enlarged, or in case of certain adenomatous conditions, when there are pathogenic lesions in the pituitary, you will find the pituitary swollen because there is a pathogenic lesion or focus in the pituitary. But otherwise, this is one gland which is extremely secured, safe, not very easily accessible, but very, very important and critical in terms of the effect that it has on metabolism and physiology of a living organism. So that's the beginning of our discussion. Let me quickly recapitulate the points. I've just said that in comparison with the sensory interventions, which happen through electrochemical signals, which in simple words is a combination of both electricity as also neurotransmitters, which operate to bring about an effect. The effect brought about by endocrine interventions requires release of hormones. And as we all know, endocrinology, the science of endocrine glands, is all about these ductless glands. And I say so because these endocrine glands do not have these specific conveying ducts. So the question obviously is, how do they ensure that the secretions, the hormones, reach the target organ? And the answer is, of course, not very difficult to understand. Imagine anything that you seek to reach your target in absence of a specific conveying duct would best be released in the bloodstream. So in course of circulation, wherever it may go and at whatever speed it may pass through, which is almost going to be commensurate with the flow of blood, it ought to reach eventually your target site. And the hormone will elicit a response only at the target site. Though it may pass through different cells of the body, the compatibility in terms of the ability to evoke or elicit a response will depend on the specific receptors which are present on the cell membrane of the target organ. And that is what is the reason that a hormone has a target specificity. Any hormone will not elicit any response in any target site. So, in terms of response and the specificity of response by the target organs to a given hormone has got to do with the presence of receptors in the right place and in the right numbers. So this is very important. Number two, sensory interventions are fast, but the effect is short lasting. Likewise, the endocrine or the hormonal controls are slower. I've already said this. It may take seconds to hours to sometimes days together before a desired effect is observed. But once the effect has been brought about, it shall last for some time. That is, it is long lasting. So I think with this discussion, let's proceed further to understand what is the importance of pituitary gland or the hypophysis in this whole triad known as the hypothalamo-hypophyseal target organ axis. A pivotal structure, so to say, in endocrinology is the pituitary gland, something very critical, something extremely important, something which is juxtaposed with the neural assembly. We talk about encephalization, which is the concentration of neurons that gives you your cephalic region within this cranium. There's something extremely important as a chief assembly of your central nervous system. You've got the brain, and on the floor of the brain, probably just beneath that, you've got a very small, tiny structure, which is known as a pituitary gland. And in all probability, the fact that the pituitary is in the proximity of the brain, and the brain has always been celebrated as the central processing unit or the CPU of a human body, or for that matter, any living organism, because that is where the signals eventually are received, processed, and prioritized for the purpose of issuing responses. So, probably people thought in the beginning that the fact that pituitary is also very close to the brain, it must be the master gland or the queen gland. So this, the erroneous, celebration of the pituitary as a queen gland, or crowning of the pituitary as the master of the endocrine orchestra, is something that needs to be revisited. And that's basically what I'm trying to talk about. I want to say this right in the beginning, that people are still divided on whether pituitary is functionally autonomous, and therefore very rightly the queen gland or the master gland, or whether it does not enjoy a functional autonomy. Rather, it in itself is under the command of something else that controls its activity. And obviously the reference is to the hypothalamus. That's exactly what we will be discussing as we run through this lecture. But I think it would be very interesting to understand why all this tiny structure was termed as the pituitary gland. So, the etymology is how you coin a particular word. So the word pituitary has Latin origins. The Latin word pituita, which means phlegm. And in the beginning, the anatomists believed somehow, I mean, that again was a fallacy. It wasn't true. It was more of an imagination, that they thought the pituitary has got to do with removing phlegm or mucus from the brain, which of course is today we look at this as something extremely ridiculous, an idea. But at some point of time in history, when anatomy wasn't progressed much, this impression gathered grounds, and the anatomists resolved to call this tiny little structure very safe and secure. And I'm going to talk to you about where exactly is the position or location of the pituitary under the the floor of the brain, safe in the depression of a particular bone known as a sphenoid bone, and the depression being known as sella turcica. We'll talk about that a little later. But for now, the reason why people call this gland as the pituitary gland was simply because they thought that this gland or this structure has got to do with cleaning up of mucus from the brain, which of course was an extremely erroneous impression that they had. So the Latin word pituita actually means a phlegm or mucus. The specific construct of the pituitary is species specific. What I mean to say is it varies. Though the general anatomy conforms to a certain basic plan, whether certain lobes are a little exaggerated, they are a little larger, they are a little more functional, or some of these parts of the pituitary are kind of a little vestigial. They are not very prominent vis-a-vis the other lobes. It's all got to do with what species it is and what is the environmental niche that it occupies. For example, we all familiar with the chameleon, which is known for changing colors very fast. And we now know that this is one animal which has got a very large pars intermedia, which is again something which you find between the anterior and the posterior pituitary. So again, what I'm trying to simply say is animals which have this ability to change their body color very fast, for the reason to camouflage themselves and get indistinguished from the background, so that they are not easily noticed by the predators or the potential predators, will normally have a large pars intermedia. And in the same breath, if you are looking at an animal which is living in xeric environment, which are very arid, dry, face a perennial scarcity of water, the insolation or the temperatures are very high, you will find that the pituitary of these organisms has got a large pars nervosa or the posterior pituitary lobe. What exactly is the role of pars nervosa in xeric environments? Does it in any way contribute to conservation of water? We shall see as the discussion proceeds further. But for now, it's important for you to remember that the pituitary construct varies between species, though the general plan of anatomy is very, very similar. The lobes may differ in terms of the expands, or how big or how small or how functional or how defunct, whether they are vestige, all that would depend on what species it is and what kind of environment does that live in. Normally, insofar as our species is concerned, I'm talking about the human pituitary is slightly oval. It's more like a pea with a stalk, right? And the stalk obviously is a reference to the pituitary stalk or the infundibulum. It's oval. That's why I choose to call it as a pea on a stalk. Weighing not more than 500 milligrams is a normal adult human being to a maximum of 1 gram. I've already said this, that during gestation, you find incremental addition of weight in the pituitary. The dimensions of the pituitary is about, say, 12 millimeters transverse and about 8 millimeters in the anterior posterior diameter. I mean, longitudinal is about 8 by 12. Well, this is exactly the question I sought to raise. If you choose to, I mean, most of us have been for an orchestra at some point of time or another. I'm just giving you a kind of a comparison or maybe a good analogy, that if you've ever seen people, playing at the orchestra, you've got a lot of musicians playing different kinds of musical instruments, but at the end of it, there is somebody, one person who stands in front and makes sure that everybody is, tuning in with one another. They play in sync, they play in tandem to ensure that they create and produce music, something very melodious to the ears. I mean, just imagine if each one were to play a tune different from the neighbor, we would probably end up getting cacophony or noise rather than music. So, I mean, that's basically what I'm talking about. That no matter how diverse and how many musicians you have in your band, and the most sophisticated and melodious instrument that you've invested in, you need a conductor, you need somebody to conduct the orchestra. So the question that I'm raising here is the hypophysis or the pituitary the conductor of endocrine orchestra? In the past, I've used the word the master or the queen gland. Now I'm choosing the word the conductor of endocrine orchestra. And by orchestra, I mean something that happens in the body to ensure that there is a synchronization, that there is a hand holding between different physiological activities, and metabolic processes are optimized, they are synchronized so that we all enjoy good health. And that's possible only when you have a good balance within you and outside you. I mean, your inside has to be in sync with your outside. And by your inside, I mean, just as we have an external environment, we also do have an internal environment, which is what is called as the inner environment or the internal milieu. And there is a Greek word milieu interior, which is basically a key to a free and independent life. Please understand, the concept is homeostasis. In course of evolution, species attempted to redeem themselves, or maybe say exempt themselves, get themselves free of their dependence on the environment. I'll just give a simple example. For instance, you talk about thermoregulation as a homeostatic mechanism. The ability to maintain a constant body temperature is not seen in absolutely all the groups of fauna, I mean the animals. It's something which is very specifically limited probably to the birds and the mammals. If you look at the reptiles, reptiles are ectothermal. I mean, they kind of depend on the environmental sources of heat to probably, gain heat, or maybe they have ways to lose it. Just give me a minute, please. Oh, apologies. So, at the end of the day, it's important to understand that organisms have attempted to invest in mechanisms that will ensure or guarantee fixity or constancy of the internal milieu, which will not allow processes in the body to go haywire and violate those ranges. I mean, that's something very important. The ultimate design is to free ourselves from the dependence on the environment in terms of processes that go on in our body. So if I am able to allow a certain physiological activity that goes on in my body independent of the environment, which is to say that I'm able to maintain a constancy of my internal environment, that is exactly what I mean by a homeostatic mechanism. And we've got a lot many homeostatic mechanisms as an investment towards maintaining constancy of our internal environment. And that's where endocrinology has a role to play. I think that's being more autonomous, is being more energy efficient, is being more optimal in terms of resource use, I mean metabolic resources. I think this is the concept for by French physiologist Claude Bernard. That's the concept of homeostasis. And the whole discussion about endocrinology needs to be viewed in the light of the need to have a constant, by constant I mean something that will not vary in wide range. A little bit of up and down is bound to happen, that these are adjustments. I mean, for example, whether it's the winter, the summer, or the rain, I think the body temperatures will not fluctuate in, very significantly. Whatever little change would happen in terms of temperature shifts will only be towards an adjustment. And that's basically what I mean by maintaining a constancy or fixity of internal milieu, which is where the pituitary, the hypothalamus, and the target organs have a role to play. The point that I've just discussed with you is the physiological homeostasis, which is the same as music that the various endocrine glands create in a body if they have a proper control and coordination by something above, which is the hypophysis, and something still above the hypophysis, the hypothalamus. So please understand, when we speak about the hypothalamo-hypophyseal target organ axis, we're actually talking about the involvement of the neural component, which is the hypothalamus, the endocrine component, the hypophysis, and of course the endocrine gland, which vary from, let's talk about the thyroid, the parathyroid, you've got the pancreas, you've got the adrenal, you've got the gonads, you've got the pineal, and the whole range of endocrine structures. So this is again very important. And let's go straight into understanding the anatomy of the pituitary. I've got a sketch here for you. It's a very simple sketch, and I'm not very sure if you're able to see my cursor, but this is more or less the hypothalamo-hypophyseal. The targets, of course, are not within the cranium. They are scattered all through the body. To start with, let's quickly understand where exactly is the location of the pituitary. Is it strategic? I mean, the way the pituitary is located in the body, more specific, or rather grossly in your cranium, the floor of the brain, at the base of the brain, you have a bone. It's a bone of your cranium called as the sphenoid bone. And in that sphenoid bone, if you see here, this bone here, which I've drawn, is a sphenoid bone. And the depression within the sphenoid bone, which takes the shape of a horse saddle, is known as sella turcica. I draw your attention to what you see on your screen, the written matter. If you see, I have put in bold words, font, sella turcica, again a Greek word, which means a Turkish horse saddle. They say again that the shape of sella turcica, the depression within the sphenoid bone of the cranium, reminded the anatomist who named this part as a sella turcica of a horse saddle used by the Turkish army men. The seat or the saddle which they put on a horseback probably had a shape very similar to this. And therefore, they named this depression in this, I keep on repeating, so that it imprints properly. I've said this in the beginning, the pituitary being so important and critical cannot be very vulnerable and accessible. It has to be very safely and securely, placed, sheltered. Where exactly you place it? You place it in the depression of the sphenoid bone, which is a part of the cranium, the depression known as sella turcica, and the specific location within that depression where the pituitary hangs by virtue of the infundibulum of the pituitary stalk is known as the hypophyseal fossa. The entire, the hypophyseal fossa is the most inferior part of the sella turcica, right? This depression, I'm not very sure if my cursor can be seen, but that's not very important. You can very clearly, because I put labels everywhere. The grayish, kind of spongy tissue that you get to see is the sphenoid bone. Within the sphenoid bone, you see a depression. In that depression, you see a bilobed structure. I've labeled it very clearly, anterior pituitary and the posterior pituitary. So this region of the sphenoid bone within which the pituitary resides is known as sella turcica. And very specifically, the most inferior part, the lowest part of the sella turcica, within which the pituitary is located, is known as the hypophyseal fossa. I have already said this before, but let me repeat. The pituitary almost seems to be a pea on a stalk. It's like a small pea, which is, held by a stalk. In this case, of course, the stalk or the funnel-shaped structure that you see going up or maybe, going downwards from the hypothalamus is also known as the infundibulum or the pituitary stalk, which is what is the connection between the pituitary, the endocrine fraction, with the brain, which is the neural fraction. So please understand that the function of the infundibulum or the pituitary stalk is to connect the pituitary and keep it in unison, in communication, and I would say, in constant physical proximity with the hypothalamus. The human pituitary is about 13 by 10 by 6 millimeters, which is, the dimension. And the weight is around, a normal adult human being has a pituitary weighing about 500 milligrams. But in certain instances, both physiological states like pregnancy and pathological states like neoplastic growths or maybe adenomatous pituitaries, you do find an increase in the weight of the pituitary. I think I put it down there. Acromegaly, for instance, I think most of you would be familiar with this condition called pituitary gigantism, or you very unfortunately label these people as pituitary giants because they've got some extremely anomalous and extraordinary height, almost between 7 to 8 feet tall, which is not a normal height for the commoners to achieve. And that's because of hypersecretion of growth hormone, which manifests into lengthening of the long bones like your humerus and eugophyma. The epiphyseal plates have not still mineralized and they keep on growing and giving a tall stature to the person concerned. So I think in these subjects, the acromegaly is slightly different from pituitary gigantism. That normally acromegaly would, if you look at the word acromegaly, acro would mean extremities and megaly would mean enlargement. So in patients afflicted by acromegaly, one would have, I would say, enlargement of digits, macroglossa, which is a large tongue. They've got a very prominent supraorbital ridge. They've got something called the bulldog scalp, which is the skin folds in the scalp thicken. They've got the nasolabial crease is very prominent. And they've got splanchnomegaly, which is enlargement of the viscera in their body. They've got cardiomegaly, which is the enlargement of heart. Of course, that's nothing normal. That's something extremely abnormal. They have a short lifespan. They're invariably diabetic. They have a lot of neurological disorders, depending on whether the pituitary is pressing on the optic chiasma or not. They can have a lot of visual disturbances. They may have double vision, blurring of vision. They have a constant headache. The glycemic index is very high. So, all these features put together, you have this condition called acromegaly, which is an endocrine disorder attributable to hypersecretion of the growth hormone. Let's now continue the discussion further. In the previous slide, I've just told you the location of the pituitary, which I said, of course, is safe, secure, strategically located within the hypophyseal fossa of sella turcica in that depression of the sphenoid bone, which is the bone of the cranium under the floor of the brain. That's very strategic. Now, I would like to speak a little bit about the zonation or the lobes of the pituitary. Very simply put, if you look at this sketch that I have drawn, it's very simple, very grossly or broad. If you look at the pituitary, it shows presence of two lobes. Going by the orientation of the pituitary, if you face in this direction, obviously the lobe of the pituitary facing the direction in which you face is the anterior lobe, and that which is exactly opposite is called the posterior lobe. And you've got technical terms. The anterior lobe is also called as adenohypophysis. This has got nothing to do with adenomatous pituitary. Please do not get confused because of phonetic similarities in the word adenoma and adenohypophysis. That's not what it means. Adenohypophysis is anterior pituitary, and I'm going to tell you the further divisions of the adenohypophysis. And then, of course, you've got the posterior pituitary or the neurohypophysis. In this sketch, I think I have put the word adenohypophysis and neurohypophysis in different font colors. So everything that is in the font color and neurohypophysis is together the assembly of neurohypophysis, whereas in adenohypophysis, you've got again many parts. Like for example, there is a kind of a collar just around the infundibulum, which is what is known as pars tuberalis. Then you've got pars distalis, which constitutes the major chunk of the adenohypophysis or the anterior pituitary. And of course, you have the pars intermedia, which I told you in the beginning, that in some animals, for instance, an animal like the chameleon, or some people call the chameleon, which is known for its amazing ability to change its skin color. In that, it would be very active and large because it's got to do with melanocyte stimulating or MSH, or it's got a hormone, it's got certain MSH secreting cells that will influence the diffusion of melanin in the skin pigments. So as I've always said this, I repeat and kindly pay attention to this because this is important, that the three subdivisions of adenohypophysis or the anterior pituitary from anterior to the posterior region are pars tuberalis, which forms a kind of a collar around the infundibulum or the pituitary stalk. The majority or the major part of the adenohypophysis is the pars distalis, which is where you have a cluster of the secretory cells. You know, there are different cell types. I'm going to talk to you about this a little later. And then, of course, you've got a very, kind of virtually vestigial, I'm talking about the human pituitary now, a lobe which is positioned between the anterior and the posterior lobes of pituitary in an intermediate position, and therefore it's called as pars intermedia, which is probably very prominent in the fetus and the newborns, but as you progress on your life track in normal adult human beings, pars intermedia is not very distinct. Of course, you do also have the pituitary has a sort of a duality of origins, outpocketing of the Rathke's, ectoderm and the neural ectoderm. So the fact that the Rathke's pouch, which is an oral ectoderm, has something to do in terms of contributing to the pituitary eventually, leaves an impression somewhere on the adenohypophysis in form of something known as Rathke's cleft, which is what you see in the diagram. So again, that's something very important. Rathke's cleft, anterior pituitary or adenohypophysis. This is the pars intermedia, just kind of between the anterior and the posterior lobe, and then you have the pars nervosa or the posterior lobe. I hope that's clear to you. So this is again very interesting. The adenohypophysis, I need to emphasize, is the largest part of the hypophysis. The majority of the cells which produce different hormone, the pituitary hormones that influence the activity of the endocrine glands are housed within the adenohypophyseal pars distalis. And we're going to talk about that a little later, that there are at least around 5 to 6 types of secretory cells, as also a non-secretory cell, which is known as the folliculostellate cell or the FS cell. But for now, what is very important for you to remember is the zonation of the pituitary. Let's start from top to bottom. You have the hypothalamus. Then you have a narrowing down, which continues further as an infundibulum or a pituitary stalk between the optic chiasma and the mammillary body, and starts entering the depression of the sphenoid bone, which, as I've told you earlier, is known as sella turcica. And the lowest part of that sella turcica, the most inferior part of the sella turcica, a depression in that sphenoid bone, which resembles the Turkish horse saddle, is known as the hypophyseal fossa. So this is more or less how it goes. And you do also have in the pituitary a lot of endothelial cells, which are vascular elements. You do also have some very interesting cells called pericytes. Let's move further. How did people start classifying the pituitary cells? To start with, it was very confusing because everything looked very, very similar. It did not really look as if the pituitary had different cell types. Which of those cells were secreting pituitary hormones, which were the non-secretory cells, whether there were some scavenging cells, it was all a confusion. So our understanding of the pituitary cytology has been gradual. It has probably taken, I don't know, several decades until the present time when we've got different kinds of sophisticated techniques and approaches to identify the pituitary cells. The simplest of all is what you see on your screen on top left, where the cells of the pituitary very categorically show differences in terms of the affinity for stains. I mean, you would notice there are three kinds of cells there. You've got chromophobes, which mean they simply do not take up any stains. Phobia, they have a phobia for pigments. Therefore, you call them chromophobes. And you have got some cells which only take up acidic stains. So you call them as acidophils. And you've got some stains which you see on top of that slide, which take up basic stains. So you call them as basophils. Then a little later, people came up with specific staining. I mean, like the PS, orangey staining, you had this staining affinity continuing into specific stains, which was one approach to identifying the cells of the pituitary. A little later, probably somewhere in the 70s, with the advent of electron microscopy, and as you would understand, electron microscopy is something which would allow you to delve deeper into the cell, not only restricting your examination and study of the tissue, but now you are able to understand the precise distribution of cell organelles within the cell, the cytoarchitecture, as you call it. And by cytoarchitecture, I mean the specific arrangement of cell organelles and cell inclusions within the cell, which imparts to the cell a typical cytoarchitectural signature. And therefore, a lot of people, started looking at cells through the electron microscope using transmission electron microscopy and were able to correlate the specific arrangement of cells with their function. And that came into force as an ultrastructural approach towards classifying the pituitary cell. Then, with the advent of immunology, where the central dogma is that you have antigen antibody specificity. So people started combining electron microscopy with immunology and started deploying certain antibodies which were tagged with radioisotopes. So obviously, if you have a slice of a pituitary which you dip in a cocktail of stains, where each stain is, maybe it has a radioisotope, because in the sense it can give you some sort of a fluorescence in that stain slide, and you tag it with a particular antibody, because the stain in this case is an antibody tagged with a radioisotope. Right? I mean, if you can see on the bottom extreme right, you see fluorescence. That is a section of the pituitary. Some bright fluorescent pink in some places, which is exactly where that particular stain, which is nothing but a combination of antibody with a radioisotope, has gone and attached to certain cells within the pituitary. Now, if we go by the logic and the diktat that the antibody shall be specific to antigen, so if I deploy, let us say, an anti-GH antibody which is tagged with a radioisotope, it will go and stick only to the secretory granules within the growth hormone cell, and that is where you will find this kind of fluorescence, or how would I say, the glow that these cells will give you, because it has got a radioisotope which has been tagged and coupled with an antibody which is the same. Why this antibody will go and only stick to GH, or rather what will go and stick to GH? What will go and stick to GH will only be an anti-GH, which is an antibody. GH. Now, how antigen antibody specificity? It has got a radioisotope. So this approach in which you combine immunology, immunofluorescence, and ultrastructure was the next level at which we started identifying the pituitary cells, and that is what was known as immunoelectron microscopy, basically a hybrid approach in which you combine immunology with electron microscopy. Of course, there was also a lot of experimental manipulation where people use certain drugs and certain surgical procedures where endocrine glands or the target glands were surgically removed, and they correlated the removal of these glands with a change in the cytoarchitecture of certain cells in the pituitary. I'll just give you an example. For instance, if you remove the gonads, total gonadectomy, which is removal of, let's say, gonads, maybe ovariectomy, or I mean, it's also called as oophorectomy, or you go for orchidectomy, which is removal of the testes, bilateral or complete, or maybe unilateral is not going to give you any, I mean, change per se in the cytoarchitecture, because you still have one gonad left that can undergo something called as compensatory hypertrophy and make up for the one that has been removed, right? So a complete or a bilateral or is more appropriate, subjected to experimental manipulations or surgical ablation, whereby you've removed, let us say, a gonad, then you find something called as a castration cell within the pituitary. I mean, the gonadotropes or gonadotrophin secreting cells, you have two of them, FSH and LH, or follicle stimulating hormone secreting cell or luteinizing hormone secreting cells, also called as folliculotropes or luteotropes. Within this, you will find some distinct changes brought about because of castration. So whatever change in cytoarchitecture that you notice following a procedure of castration or surgical ablation of a target site. Now, why am I saying target site? Because the target for follicle stimulating hormone and luteinizing hormone, of course, is the gonad. What am I doing as an experimenter? I'm removing the target site. So, in my opinion, through a feedback mechanism, the fact that I've removed a target site should cause some sort of a disarray or disruption of communication along the hypothalamo-hypophyseal target organ axis. That is the logic. So this is a sort of a reverse, how would I say, endorsement or confirmation of what cell has a role to play with which target site. I hope that is clear to you. I mean, I've made an attempt to put it in very simple words. So and of course, you also have something called parabiasis. If you see this, I'm so sorry, apologies. If you see this particular, diagram where you have two rats or two mice or two whatever mammalian models that have been, sort of fused with each other, that the vascular supply has been continuous. Let me explain what this means. Parabiasis is a procedure in which you connect the vasculature of one animal which is with another, and they both obviously they are conspecific and they have tissue compatibility. I mean, both are mice, they both belong to the same species, so the anatomy is similar, and there is very little chance that there will be some kind of tissue incompatibility and rejection. So, point very simply is, if you surgically manipulate or experimentally manipulate one of these two, you shall also find a similar effect that you expect here in the other animal. Why? The reason very simply is because they have the same vascular supply. And the vascular supply, I mean to say, blood is the portal through which the hormones flow. So if I manipulate something in, let us say, of the pair of parabiased animals, I manipulate something in A, I should also get an effect of similar, intensity or strength in the other animal with which this has been parabiased. By parabiasis, I mean you connect the vasculature of these two animals and try to do your permutation combination experiment. So this is basic. And last but not the least, in the center, I've that's what I told you. I have put those images. I mean, these are all borrowed images. I must uh, tell you that this kind of work is now not possible in undergrads, and I mean, you need a specific permission to use laboratory animals. You need to have a dissection monitoring committee to oversee the entire, operation. But however, as and when and as and where this is possible, if you remove, for example, any of those target sites, I remind you of the whole discussion. We have been speaking about hypothalamo-hypophyseal target organ axis as a triad, and the communication coming from the higher centers or the neural centers in the brain, which is the hypothalamus, down into the pituitary, which stimulates something in the pituitary, either in the adenohypophysis or the neurohypophysis, further, eliciting release of those pituitary hormones into the blood through circulation. Finally, it reaches the target site and brings about whatever the desired effect is. So this is basically, this slide was primarily to explain to all of you the approaches taken for classifying pituitary and the milestones that we have, crossed in the last whatever number of decades that people have been researching the pituitary, starting with something very basic in terms of whether pituitary has got cells that accept the stain, and if they accept the stain, whether the stain that they accept is acidic or basic, accordingly you call them acidophils, basophils, and then you do also have a population of cells where they don't seem to be, showing any sort of affinity towards stains in the sense that they are chromophobes. So this is again very important. I would like to spend a couple of minutes explaining to you the cytoarchitectural features or ultrastructural peculiarities of adenohypophyseal lactotropes. This is my personal research work, and I thought this would be very pertinent for me to share this with you. If you recall, a few minutes ago, when I was explaining to you the schemes for classifying pituitary, I did make a mention that one approach by which you can classify the pituitary cell types, particularly of the adenohypophysis, please remember, if you recall a slide which was showing you the zones or maybe the lobes of the pituitary, I did speak to you about adenohypophysis and neurohypophysis. Further within the adenohypophysis, I did say you've got three subdivisions: pars tuberalis, pars distalis, and pars intermedia. And then I emphasized that you've got as many as 5 to 6 secretory cell types within the pars distalis, which is a major constituent of adenohypophysis. Please understand and remember this. That the cells like growth hormone cells, you got somatotropes, you got lactotropes, you got corticotropes, you got thyrotropes, you got gonadotropes, which again are folliculotropes and luteotropes. Where exactly are all these cells? Of course, these cells are constituents of adenohypophyseal pars distalis. And among all these cells, my personal favorite and also the cell by most of the endocrinologists for the kind of receptivity and sensitivity to any sort of manipulations that these cells show you. The most I would say interesting among all these six secretory cell types is what is known as adenohypophyseal lactotrope or prolactin secreting cell. Now, the what you have on your screen, which I am presenting to you, is a lactotrope of a rat. Rat is norvegicus, all right, a laboratory rat. The one on the left is a normal prolactin cell. It has its own interesting cytoarchitectural feature. It is angular. It can also be stellate, a star-like appearance. You notice a very prominent nucleus with more of euchromatin than heterochromatin. And may I draw your attention to two important points on the left-hand side. There are two images. One on the left is a normal. The one on the right is a prolactin cell in a state of high synthetic activity, such as that which may be encountered in the pituitary of a pregnant woman. So if you very carefully look at the cell on the left, you will notice the ergastoplasm or the rough endoplasmic reticulum is arranged in, typical staked arrangement. It's a kind of parallel stake of cisternae, right? Which are swollen and whatever. Not so really, I mean, it's very normal. And if you look at the secretory granules, which appear very dark, right? This is exactly where the secretion of the prolactin cell or prolactin secreting cell or lactotrope. By the way, all of us know prolactin is a lactogenic hormone which plays a critical role in lactogenesis in anticipation of postpartum responsibility of the maternal organism for nourishing the newborn fetus. So here, if you look at the morphology of these secretory granules, you'll find that they are pleomorphic, which means varied morphology. And by varied morphology, I mean they don't conform to one definite shape and one definite dimension. They're very varied. Now I draw your attention to the image on the right, which is where you see some very interesting designs. And this is a slightly, at a higher magnification electron micrograph, where one thing I specifically want to draw your attention to is what I have circled in the red. You very clear, and this is a part of my own research and doctoral research, and you will postdoctoral research too. And you will very clearly notice that the manner in which the cisternae of the ergastoplasm or the rough endoplasmic reticulum, very, very obviously it is rough endoplasmic reticulum in both the slides, because you see the cisternae are studded with ribosomes, right? There's no doubt about it. But are you able to appreciate that the cisternae, unlike in case of the normal cell, where they are in parallel stakes, in this case, they are arranged in concentric rings. So the question that one obviously is confronted with is, why this sudden shift in the manner in which these endoplasmic reticular cisternae suddenly go from parallel stake to concentric rings? And the answer is obvious. If you want to accommodate a greater length, the more easier way to do that is arrange it in a concentric manner rather than in a straight parallel stakes. So it implies that in this particular organism, in all probability, there has been a reason as to why the synthetic activity or the status in terms of synthesis of prolactin hormone, because we're talking of prolactin cells, is heightened. And obviously, as I've told you, all through gestation or pregnancy, with advancing stage of pregnancy, the lactogenic function is also proportionately heightened. Now, what do you need to support lactogenesis, which is milk production in anticipation of a maternal function in the postpartum phase? By which I mean, once the fetus is delivered, then the reflex arc will be instated and you have a milk letdown, which of course has nothing to do with pars distalis. Yes, pars distalis. That is a function of oxytocin producing cells in the pars nervosa or the neurohypophysis. But what is very interesting, and this is something beautiful, this is all how it unfolds as the milestones of gestation are reached. You have ever increasing more and more ergastoplasmic elements appearing in the cytoplasm of the lactotrophs, which keep on forming those concentric rings in the cytoplasm of the cell, and that's what is known as a Nebenken formation. That's the word. Whenever, I repeat, this is important, that whenever you have a cell in a state of heightened synthetic activity, uh, obviously there would be a need for correspondingly higher endoplasmic reticular cisternae, because Golgi endoplasmic reticular cisternae is the place where you have all this protein synthesis, right? So if the cell is expected to synthesize and secrete more, I mean, if it is to secrete more, it has to synthesize more. If it has to synthesize more, then obviously it has to accommodate more, the greater lengths. So this is the design. So coming to the point, any cell in the pituitary which shows the presence of Nebenken formation is 100%, undoubtedly, you can close your eyes and say this is a prolactin cell. Now, this is exactly the approach known as cytoarchitectural features. This is a signature feature of the prolactin cell. And you can say, I mean, those of us who have access to electron microscope, I think in most of the universities today, we have electron microscopes. It's a slightly expensive technique, but it's well worth it. That if you are able to delve into the cell and not just appreciate cells as a cluster, I mean as a tissue per se, and get into understanding the cytoarchitectural peculiarities, I think you will be doubly sure as to what is the identity of the pituitary cell. So again, this is very important. And you may ask me, is it only got to do with the prolactin cell, or does every single cell of the pituitary has a cytoarchitectural feature? Well, the answer is absolutely every single cell will, how would I put it, have something very specific in terms of cytoarchitecture. For example, the growth hormone cells are the most abundantly packed with secretory granules. For example, if you look at the adrenocorticotrophs, ACTH secreting cells of the adenohypophyseal pars distalis, then you find something very amazing, that the secretory granules in the adrenocorticotrophic cells will line up in a single file under the plasma membrane. I mean, that is the beauty. I mean, you will not find this with any other cell. So as you are examining these samples under the electron microscope, and the investigator comes across a cell or a cluster of cells where you have in each cell a number of secretory granules lining up in a single file under the cell membrane, I mean plasma membrane, you can be absolutely sure that the cell that you're looking at is the adrenocorticotrophic cell. I've already explained that to you. Then for gonadotrophs, you normally have a juxtanuclear Golgi zone. So, I mean, in simple words, again coming back to the point, whenever you rely on this kind of ultrastructural peculiarities of, the adenohypophyseal pars distalis cells, the cells of somatotropes, thyrotropes, adrenocorticotropes, lactotropes, gonadotropes are of two types. You have the LH cell or the luteotrophs, and you have the FSH cell. I'm sure we all know this, that when it comes to the folliculotrope, that's got to do with secretion of follicle stimulating hormone that is implicated in gametogenesis. And if you're looking at the luteotrope, that's got to do with release of the LH or luteinizing hormone, for which the target organ is, of course, the corpus luteum, which is the empty-graphene follicle in the ovarian tissue. After ovulation, the empty-graphene follicle will pass through a number of phases like corpus hemorrhagicus, and you've got corpus luteum, and sometimes you do also have the corpus albicans, and then you also have adratic follicles. But a fully formed corpus luteum, which is a proper glandular, structure, will be the most critical source of a hormone which is required for maintaining pregnancy without causing a miscarriage. And that's, of course, the progesterone hormone, which is secreted by the corpus luteum. So, I mean, these that you see on your screen, I'm not going to get into the specific details. That's not in the scope of my discussion. But what I want everybody to remember and appreciate is that every single cell type of adenohypophyseal pars distalis, in simple words, the anterior pituitary lobe, has its own structural or cytoarchitectural features and peculiarities based on which it could be identified. All these, as I said, there are about 6 of them. Sometimes people only talk about gonadotropes as a single entity, but we know we have and before I forget, you do also sometimes have confusing situations in which a single cell type is responsible for a duality of hormonal output. For example, you got mammosomatotrope, which is a combination of mammotrope, which is a lactotrope, plus a somatotrope or a growth hormone secreting cell, which means the same cell shall be the source of 2 hormones. And sometimes it is more than 2 hormones as well. So this is very interesting. And this is a phenomenon which was probably realized when people combine immunology with electron microscopy in a bid to, be more specific. I think I've already explained that to you, that not only to use antibodies, but use radioisotope tagged antibodies as your stain. I mean, you use, and it'll go and actually show fluorescence only specifically in the cells for which it has a compatible antigen. What is the antigen? The secretion. And what is the antibody? Antibody is what you have tagged with radioisotope and you've added it to your stain. So, when one single cell is showing fluorescence for more than one radioisotope or antibody tagged stain, this is where not just one but multiple hormones are coming from. So this concept is called as plurihormonality, which means not just one. I mean, this is something very interesting. And this is not very difficult to understand because there is always a stem cell reserve in the, in the pituitary, that depending on the physiological state of the body and depending on the urgency and the need for certain priming, or in anticipation of certain critical functions that the body is going through, as I've said. I mean, in a pregnant subject, there is so much upheaval, it's kind of, how would I say, a lot of anticipatory exercises that go on in the body in preparation for motherhood. Whether it's got to do with, I mean, as you near the full-term pregnancy, there are going to be definitely a lot of changes in terms of your pubic symphysis in anticipation of formation of birth canal. There is going to be engorgement of the mammary tissue and filling of alveoli with milk, but it won't be released, of course. Milk ejection is a different story altogether, and that is under the control of the second smaller lobe of the pituitary, which is called as neurohypophysis, or the pars nervosa, and which is where again you have 2 specific cluster of cells, which is what I'm going to quickly go through, known as supraoptic and paraventricular nuclei, which are responsible, of course, for oxytocin and vasopressin. So even when it comes to feeling thirsty, the thirst, that sensation where you feel I need to drink water, I need to have some fluids, is something which does not arise here. Thirst is not here. The thirst is a sensation which arises here. And that's basically the hypothalamic cluster or osmoreceptive when, a cluster of cells which are extremely sensitive to fluctuating osmotic pressure of your blood, that a blood which is passing through that critical region called as the thirst center of your brain, if has a higher osmotic pressure, because the concentration of salts is more than the water potential. See, it's a basic concept in physical chemistry. If you got a solution, it ought to have a solvent and solutes. And the osmotic pressure of that solution will depend on the ratio of solutes to solvent. If you got more water, then the solutes, the osmotic pressure is low. If you got more water than the solutes, the osmotic pressure will be lower. These fluctuations on a regular basis, the osmoreceptor cells of the hypothalamus, that cluster, which is what constitutes your thirst center, is able to sense. And accordingly, it will switch on or switch off. And this switch on, switch off mechanism of the thirst center will operate through releasing another critical, secretion coming from the hypothalamic cluster. It is not produced in the pituitary. Rather, it is only brought down along the axon transport and stored in the neurohypophysis or the pars nervosa and released in response to this kind of sensations of thirst. And what am I talking about? I'm talking about the antidiuretic hormone or the vasopressin, which is again a fantastic molecule which makes sure that you do not get dehydrated by losing a lot of water through the urine. I mean, you have polydipsia, which is you frequently, you have polyhedra, polydipsia, you've got frequent urination. You've got diuresis, which is passage of dilute urine. I mean, if that goes on unchecked, I'm sure you agree with me, the body would be dehydrated and less water is going to cause a lot of problems, right? So it can cause a metabolic arrest and everything will go haywire. So, I mean, the criticality of the pituitary, whether it's the adenohypophysis or the, neurohypophysis, is absolutely unquestionable. Let's continue further. And this is basically what I was talking about. I have put a very simple sketch for you to understand the communication. Again, please let's go back to what I started with. We're talking about the hypothalamo-hypophyseal target organ axis, which means communication happening between three critical, crucial, discrete components of this assembly, which ought to function in unison by holding each other's hand. If they do not do that, things will go wrong. And to ensure this sort of synchronization, that they work together towards the larger good of the organism in terms of maintaining a fixity or constancy of the internal milieu to ensure homeostasis, what needs to be done is proper communication channel. If I am to keep good rapport with my neighbor, I need to communicate. If I am to have cordial relationship with my colleagues, I need to communicate. Often, we say this, that lack of communication creates this sort of a rift. And it's no different with hypothalamo-hypophyseal target organ axis. They need to communicate properly and they need to communicate with precision. And that's possible. How is it possible? As you can see from this very sketch that I have drawn for you, that if kindly look at the hypothalamic part, which is right on top, right? You can very clearly see here that the hypothalamic part in between a little constricted, sort of a funnel-shaped infundibulum or the hypothalamic pituitary stalk. And then you have this, as I call it, a pear on a stalk, right? So if you look at the hypothalamic region, I have used three different colors, right? Probably, I think one is probably purple, and one is a little green, and one is blue. May I draw your attention to the first thing, which is a cluster of arcuate nuclei. This is very important. Basically, you've got parvocellular neurons, which also is where you find the arcuate nuclei located in the hypothalamus, close to the mammillary body. Very important, I repeat, the arcuate nucleus, in simple words, are the cyton or the body of the neuron, and it's a cluster from where you will have certain regulatory molecules or hypothalamic releasing factors that will come down along the axon. And if you see very carefully, I put a circle here. The hypothalamo-hypophyseal tract is nothing but a bundle of axons arising from the neurons, or to be more precise, the cyton or the arcuate nuclei will terminate somewhere here. It doesn't continue further or end up in the pituitary at all. Whereas if you look at the other two clusters, which is the paraventricular hypothalamic nuclei and the supraoptic hypothalamic nuclei, you very clearly notice that they continue further down. The axon bundles do not terminate halfway through, just like the axon bundle of the arcuate nuclei. They're continuing further down into the neurohypophysis. So that the axon bundles, of which you have two discrete sets, one being paraventricular hypothalamic nuclei and the supraoptic hypothalamic nuclei, arising from a part of the hypothalamus which is more close towards the optic chiasma than the mammillary body, will traverse through the hypothalamic stroma and come down through the infundibulum right into the neurohypophysis. Unlike what is seen with arcuate nuclei, that the axon terminus abruptly terminates in a small region, which is where, as you would see in the next slide, the vascular component of the hypophyseal portal system exists. I think I'll just put that on hold. At this sketch only tells you the communication portals between the hypothalamus and the pituitary. That in the hypothalamus, you got three clusters of neurosecretory cells. One known as the parvocellular, the other known as magnocellular. Magnocellular further categorized as paraventricular and supraoptic. Parvocellular has got to do with arcuate nucleus. Number one. The arcuate nuclei are responsible for secreting certain regulatory molecules, which is what are called as hypothalamic releasing factors as well, which are brought down along the axon transport. By axon, I mean to say the axon bodies of all those neurons which arise from the parvocellular neurons are also known as the hypothalamo-hypophyseal tract. But contrary to this design, if you look at the arrangement of paraventricular and supraoptic optic hypothalamic nuclei, they do not abruptly terminate halfway through. Rather, they traverse through the infundibulum and terminate, end up with neurohypophysis. That's the beauty of it. What does that mean? In all probability, it means that the neurohypophysis actually is only a storage site. It does not have any secretory components. Please be very clear about this, that there is no parallel or counterpart of the secretory cell cluster that you see within the adenohypophyseal pars distalis, of which we have just had a discussion. I told you about 6 cell types: somatotropes, lactotropes, thyrotropes, adrenocorticotropes, folliculotropes, luteotropes. These are all secretory cells. Plus, if you recall, even within the adenohypophyseal pars distalis, you have a folliculostellate cell, which is a scavenging cell. It is a non-secretory cell. Is there that kind of a extraordinary diverse assembly of cells within the neurohypophysis? The answer is a big no. Neurohypophysis does not have anything which secretes any hormone or regulatory molecule, but it only stores what comes down from the hypothalamic paraventricular and supraoptic hypothalamic nuclei. And these are two important molecules, oxytocin and vasopressin. And that's what I was trying to explain to you. Oxytocin has a role to play in prepartum physiology as also postpartum physiology. In prepartum physiology, it has a role to play in terms of initiating labor, in terms of putting the myometrium of the uterus into a vigorous violent contraction. That's called as the initiation of parturition or break, a little after which you have breaking of the amniotic bag. And that is the sign that the fetus is gradually making its way out. Kind of relaxing of the pubic symphysis, alignment of the vulvovaginal orifice with the cervix to ensure that there is a proper birth canal for the fetus to pass through. All this happens at the behest of oxytocin in the prepartum phase, right? Whereas in the postpartum phase, it is the same molecule which will activate the milk ejection reflex arc. The myoepithelial cells in the areola of the breast, which is where the newborn will suckle at will trigger that milk ejection reflex under the influence of oxytocin for milk ejection or release of milk. So this is the beauty. Everything, our body, I think, is the most extraordinary bioengineered marvel, as I call it. We don't appreciate that because these are things which are happening inside in terms of homeostasis. What the outward manifestations are not very easy to appreciate, and until and unless something goes wrong, one would not realize what is the effort that has been put in designing our bodies. When things fail, we realize the importance, and in hindsight, you realize how complex our mechanisms are. So I think this is very important, and we move simply into the other one. This is quickly, I'm just going to very briefly talk about this. This is called as hypophyseal portal system, which simply means a vascular arrangement in which you have, it starts with a capillary and ends with a capillary. And I'll put it very simply. This is an extremely simplified schematic diagram. That a branch of the carotid artery called as the superior hypophyseal artery, you see on top, and the red arrow, the thick one which enters, that is called the entire knot of capillaries which is formed because of branching of that superior hypophyseal artery. That's called as I would say ramification somewhere near the pituitary stalk, or the the so-called infundibulum, which is where, and may I show you that previous diagram. Please note that the axon bundle arising from arcuate nuclei terminates exactly at that point. That is the primary plexus of the of the hypophysis. Please note that very clearly. Whereas after this, if I'm so sorry. Whereas it all, and then there are of course two parallel draining veins. I mean, all this knot of capillaries called a primary plexus will anastomose again to form two parallel veins or venules, if you like it that way, and they will again sort of consolidate to form the secondary plexus. These are again net of, I mean, it's a kind of a capillary net around the secretory cluster, I mean, the cluster of secretory cells. I remind you, this is the adenohypophysis, which are drawn in pink, and these cells around which the secondary plexus is forming a sort of a net, or a, sort of a network or plexus, is the same cluster of adenohypophyseal pars distalis cells that secrete, whose secretion will be going out through the anterior hypophyseal vein into the peripheral circulation, to be moved through the general circulation until it reaches the target site, which is where it will elicit a response. I have said this very clear to you. So, I mean, the billion-dollar question now that arises is, why this complex arrangement of a hypophyseal portal system? By the way, a portal system is that which starts with a capillary and ends with a capillary. The reason is very simple. The reason being, anything which is coming from the hypothalamus in terms of a regulatory influence, if it has to if it has to reach the secretory cells within the adenohypophyseal pars distalis in the least time possible and the quickest possible time, it wouldn't make sense to release that into a general peripheral circulation. By the time it circulates through the whole body and reaches again into the adenohypophyseal pars distalis to stimulate those secretory cells, prompting them to release their own secretions further into the bloodstream, under whose influence, of course, these are those trophic hormones. By the way, these regulatory molecules coming from the hypothalamus are also known as hypophysiotrophic hormones, because they nourish the pituitary and they stimulate the cells of the adenohypophyseal pars distalis to release their own trophic hormones further down into the bloodstream. I hope that's clear to you. Question is there a similar arrangement with the neurohypophysis in terms of vasculature? The answer is no. Have a look at this. It is a simple pardon me, entry of an inferior hypophyseal artery with a single plexus, no hypophyseal, I mean, no portal system. May I remind you, I have made this very clear to you. A portal system necessarily shall have and should have the starting point as a capillary tuft or a network, and ending also with a capillary tuft. Here there is one single tuft of capillary which is drained by a hypophyseal vein. So what is the reason here? The reason here is very simple. The reason here is what is coming here and what is being delivered here is already, it's a ready molecule. It need not stimulate anything in between, unlike the case of adenohypophyseal pars distalis. See, again, let me explain. that something coming from the hypothalamic, hypophyseal tract into the adenohypophysis is a hypophysiotrophic hormone. And that is not the hormone which is going to stimulate your endocrine glands. It is doing it through the agency of pituitary hormones. Whereas what is coming down, as I've explained to you, the paraventricular and the supraoptic nuclei, namely the oxytocin and the vasopressin molecules, are ready to be released into the blood because they are synthesized itself in the hypothalamus and they have to be secreted, released further into the blood. So what is the function of pars nervosa? Only a storage site, which is why you don't need any sort of a portal system here. I hope that is clear to you. These are the same details again. As I told you on the left hand side, please have a look. Magnocellular, parvocellular is the same as arcuate. These are the hypophyseal, hypothalamo-hypophyseal tract coming from parvocellular will terminate in the primary plexus, whereas those arising from magnocellular will go straight down into the posterior pituitary, right? Inferior hypophyseal artery drained by veins. Here the superior hypophyseal artery drained again by two venules. And then again secondary plexus around the pars distalis being drained further by veins. No more, not much. Quickly, let us look at the various hormones. You've got the hormones released by the anterior pituitary are, as you see on your screen, you've got the growth hormone, also called the somatotropin, which is released by the growth hormone cell under the influence of growth hormone releasing hormone. That is, plus means a positive influence. You also have somatostatin, which is an inhibitory regulatory molecule, again coming from the hypothalamus. If the somatostatin will not allow the growth hormone cell to be released by the somatotropes, the target organ, liver, muscle, bone, kidney, whatever you see there, and it normally acts only with a mediator, which is a substance which mimics insulin. It's an insulin mimetic substance called insulin-like growth factor 1, IGF-1. Similarly, thyroid stimulating hormone, you have a very specific hypothalamic regulatory molecule called TRH, or thyrotropin releasing hormone. There is no negative influence here. The target organ is the thyroid. The primary organ, the hormones released by the thyroid are, of course, you know that it's thyroxine and triiodothyronine. Similarly, adrenocorticotrophin is a hormone released by corticotropes, right? And there is a positive influence coming from the hypothalamus. The target site is adrenal cortex. Of course, we all know a little bit of testosterone, glucocorticoids, and mineral corticoids is the output by the adrenal cortex. And then, of course, I've explained to you the FSH, or follicle stimulating hormone, which push those follicles both in the ovaries and, of course, the precursor stages of a matches spermatozoa, which is the primary, secondary spermatocytes. You've got spermatogonia, of course, and spermatids, and finally the spermatozoa. All that, the sequential progressive development is under FSH. In luteinizing hormone, very interestingly, is ideally not called as luteinizing hormone in men, because luteinization does not happen there. Luteinization, as I've just explained to you, is the transformation of the empty graphene follicle post-ovulation into a yellow glandular mass known as corpus luteum, which, let me repeat, I've already said that, is a major source of progesterone for maintenance of pregnancy. Now, one would ask, what does it do in men? Because there's no luteinization happening, because there is no ovary, absolutely. So the answer is, this is a slight misnomer. It's ideally called as interstitial cell stimulating hormone, or ICSH, which has got to do with stimulation of cells Leydig, which are present between the space interstices of seminiferous tubules, and the cells are Leydig. I'm sure all of you know that there are different kinds of cells in the testicular stroma. You've got the nerve cells, you've got the Leydig cells, and you've got the germ cells, right? So from among all this, you've got the cells Leydig, which respond to the luteinizing hormone, which in men or in males is more appropriately called as the interstitial cell stimulating hormone. Of course, you've got the prolactin cell, for which you do not have a stimulating influence from the hypothalamus, but also an inhibitory influence. It's believed to be a dopamine molecule, and the target site is the mammary tissue of the breast, and it's responsible for lactogenesis. I'm not just going to go into great details. I'll just quickly run through the four hormones released by the pituitary, or the the so-called trophic hormones are the growth hormone, the thyroid stimulating hormone, adrenocorticotropic hormone, FSH, and the LH. Of course, the anterior lobe also releases thyroid. This is very critical in normal metabolism. I mean, sometimes you have a lot of sweating, shifts in your body, I mean, temperature. You're putting on weight without any obvious reason, though you aren't eating much. You feel very tired. I mean, that certainly calls for profiling your thyroid hormones. Overproduction of TSH is also called hypothyroidism. Lack is called hypothyroidism. You also have things like exophthalmic goiter, which is something to do with anomalies in the titers of thyroid stimulating hormone. You've got adrenocorticotropic hormone, excess and less of which is also not right. So, this Cushing syndrome, which is basically because of hypersecretion of ACTH. I mean, this adrenal gland is very important in terms of stress hormones, particularly cortisol. And cortisol regulates the body's reaction to stress. I mean, you talk about the flight and the fight response, right? So through cortisol as an intermediary, you have a lot of adjustments in glucose metabolism, blood pressure, which is cardiodynamics, uh, anti-inflammatory effects. So again, adrenocorticotropic hormone is important. I've already explained to you the importance of FSH, or follicle stimulating hormone and luteinizing hormone. I would not wish to repeat that again. Growth hormone, right in the beginning, I just speak to you about excessive production of growth hormone leading to pituitary gigantism, sometimes accompanied by acromegaly. I've given you the entire list on what acromegaly can do, really speaking. I mean, it can cause splenomegaly, enlargement of the viscera, macroglossa, very prominent supraorbital ridges, nasolabial crease or folds. You've got diastema, which is a gap between the teeth. You've got macroglossa, a very large protruding tongue. You're diabetic. You're cardiomegaly, which is your heart is enlarged. So all in all, the lifespan is not very big. They are diabetic as well. Prolactin, I think I did spend a lot of time explaining to you prolactin vis-a-vis Nevin conformation. That's again critical in maintaining lactogenesis. And prolactin, of course, is anti-estrogenic. And I'll just spend 5 minutes to explain to you what that means. If you ever wonder that many of these women who after their delivery have been nursing or breastfeeding their children for a long time, they kind of get covered by natural contraception because of the hyperprolactinemic state, which means as long as they nurse, it's the demand of the body to produce more milk, and for which they need to have a constantly heightened titer of prolactin in the ovarian, the primary follicles into that development. So there will be um, no what's the fertile state. No, that's something very, very important. So I believe heightened levels of prolactin definitely manifest into some kind of anti-estrogenic activity. I believe, let's quickly take a recap. I think in today's lecture, I just speak to you about the etymology of the word pituitary, its strategic location, its anatomy and zones in terms of lobes, the various approaches to classifying the pituitary, how they've evolved from simple staining affinity to something very advanced using a combination hybrid approach of combining electron microscopy with immunology. I did speak to you about the strategic angioarchitecture or vasculature of the pituitary, particularly talking about the hypophyseal portal system. I did speak to you very briefly about each cell type in the adenohypophyseal pars distalis. And last but not least, I did touch upon all of these pituitary hormones, both coming from the adenohypophysis as also from the neurohypophysis. I think I appreciate your patience, and with that, I end this lecture. Thank you very much. Have a pleasant evening. God bless.